Hey everybody, welcome back to my channel. So today we are going to discuss about enzyme kinetics, and the major topic of today's video is going to be Michaelis-Menten model. So before starting this video, I would like to tell you that this video is going to be in English. If you are comfortable in Hindi, then please switch to the video which is in Hindi. I will provide its link in the description box. So let's get started. So first of all, what is enzyme kinetics? So it is the study of the rate at which the enzyme works, or the rate of the chemical reaction that is being controlled by a particular enzyme. This is the enzyme kinetics, and its study was majorly done by two scientists, Michaelis and Menten, in 1913. And by studying the enzyme kinetics of a reaction, they also deduced a mathematical expression and showed that there are two important constants. One is Vmax and another one is Km, and these two constants play a very important role in enzyme kinetics. So what was the Michaelis-Menten model? This model was based on the study on the chemical reaction which was catalyzed by the enzyme invertase, and invertase is known to degrade sucrose to glucose and fructose. That is, it degrades the disaccharide to monosaccharides. And by studying the chemical kinetics or the enzyme kinetics of this reaction, they showed a particular relation between the velocity of the reaction and the concentration of the substrates. And they showed that when we plot a graph between reaction velocity and the substrate concentration, we get a hyperbola curve. So this is how the curve looks. Here you can see that on the y-axis there is reaction velocity V. On the x-axis there is substrate concentration. So here you can see that when the substrate concentration is increasing, the velocity of the chemical reaction is also increasing, but only up to a certain limit. So here you can see that the chemical reaction is stabilizing, it is becoming stationary. So this velocity at which the reaction is stabilizing is known as the maximum velocity, or the Vmax, and half of this Vmax, or the substrate concentration at which the maximum velocity becomes half, is known as Km. So Km is the Michaelis constant, or the substrate concentration at which the reaction velocity is becoming half. So we have studied that Vmax is the maximum rate of the reaction, or maximum velocity of the reaction, where all the active sites of the enzymes are saturated with the substrate. That is, all the enzymes have bound to their respective substrates. What is Km? So Km is the substrate concentration where the maximum the velocity becomes half of its maximum value, and lower the Km means that less of the substrate is required to reach half the maximum velocity. So what is Km? Km is also known as Michaelis constant, and it is the concentration of the substrate at which the reaction velocity reaches half of its maximum value, and this constant's value, that is Km value, varies from the enzyme to enzyme, and it depends on various factors such as temperature, nature of the substrate, pH, and also the ionic strength. Now, the Michaelis-Menten constant that we have studied earlier is based on that particular graph between the velocity of the reaction and the substrate concentration. But that graph is not very accurate, as we cannot find the Vmax of a particular reaction by that graph only. So there is another graph that is plotted to find out the Vmax value accurately, and this plot is known as Lineweaver-Burk plot. It is also known as double reciprocal plot, and this plot gives us the value of the Vmax accurately. Here you can see that it is the plot between the reciprocal of the reaction velocity and the reciprocal of the substrate concentration. And by reciprocity of these values, we can get a straight line. So this is also known as a straight line plot. And here you can see that the x-intercept here, this region denotes the minus or the negative reciprocal of the Km Michaelis constant, while this point is known as the y-intercept or the reciprocal of the Vmax. So by knowing these particular points, we can easily know the value of Vmax. So Lineweaver-Burk plot is very accurate for the for defining the property of Vmax or the Km value. There are other graphs also, but this is the particular graph that is most important. So you need to learn this only. Now, in this video, we have discussed about Km and Vmax. In the coming videos, we are going to discuss about the factors that affect the enzyme activity, such as temperature, pH, and how they are affecting the activity of the enzyme. So stay tuned. Goodbye.